Mm. Mm. Woo! That's good. Hey, squad. Cold and wintry outside, warm and sciencey inside. So pull up that computer, all of your supplies at your little workstation, wherever that may be, and let's get after some understanding about the amazingness of blood, shall we? Um, so, what we have here is our book, okay? And we started by cutting out some things, so get them all out of your pocket in the back. And we had put in the title, Blood Analysis, a little definition of forensic serology. And what I want you to do is take the little test tube where we see that we've got the parts um, of blood have been separated. And we'll talk about that in a second. And then also what I want you to do is to take this little graphic and paste it right here underneath the forensic serology. And this is a little arrow you can see right here that goes like this. Kind of showing this and this with the arrow pointing down to what we're going to be getting into here. After you do that, the next thing I want you to do in preparation for the learning is to take the supplies that you have here or the different graphics, and we're going to cut those out. And there's two ways that you can do that. You can use your scissors, or if you're using your X-Acto knife and you've been like, man, I can't wait to use that X-Acto knife, going to need your cutting board for this. You can use this. So I'll show you both ways how you do that. If you're using your scissors, some things like this would be easy to cut out because we're just going to kind of cut them out in squares. And I just cut them out into rows before separating the individual. They look like little light bulbs. Or if you're going to use your X-Acto knife and you want to get some X-Acto knife skills going on. Now, I use a metal ruler, not a plastic one, because if you're not holding the X-Acto knife perfectly straight, what can happen is it can cut into the plastic ruler. You just put the plastic or the ruler down, hold it, try to keep this as straight up and down as possible. Okay, and you're just separating out these. Okay. And once you do that, you can always be aware of where your fingers are. Remember, with the long, straight cuts, you want to lay your blade down more so you have more of the blade surface interacting. If you're going around corners, you want to tip it up more to the edge because if there's less of the blade um, in the material, it'll be easier to turn a corner versus having the whole blade down and you're trying to turn this way. And so, can go up on the top and just cut this around. Yes, cut this off here. So depending upon how busy your day is, sometimes I like to just do this because it helps settle my mind, helps get me focused for the learning ahead. Just do this little preparation. This is probably not the best use of the X-Acto knife, cutting out these little antigens. It doesn't have to be perfect like this. But where this really comes in, the X-Acto knife, I think is more helpful, is when you're separating out these. We'll learn that these are our antibodies. And so you can just separate these. And then with these, I just cut like this, like this, like this, something like that. And you can just kind of cut out the Y shape way these antibodies look. Try not to leave too much of the excess white around there. Okay. And so you would have something like that. Or, like I said, you can just use the scissors. I find having some X-Acto knife skills, it just gives you experience with another type of tool that you would use, I don't know, for making Christmas cards or crafting. It's just another tool to use for all kinds of purposes. Okay, and then with this, again, with the scissors, you just can separate them out. And then, or you can do both. Cut this out and then use the X-Acto knife to just cut out the top portion here. Lay the blade down. And I got another one like that. So go ahead and cut out each of these little antibodies and cut out all of the little light bulbs. Keep your workspace organized, so slow down, don't rush, so you don't throw away any of these, get them mixed up with the little scrap pile that you're creating there. And I will check back in with you when we are ready to go. The other thing, while we're talking about cutting things out, in my scrap pile, I've got my antibody pile. I'm going to have my little antigen pile here. So you're also going to, goes over here, here. When you're done with that, make sure you cut them all out. You're going to be cutting out these red blood cells here. So I cut them out like this. Initially, just come out like this. Okay, separate them. And then again, just use the X-Acto knife. Okay. Or what you can do is, again, with the scissors might make it a little more smooth. So you don't have so many rough edges. When you're using the scissors, come right up to where the scissors come together, and you don't want to get to the end of the scissors as you're cutting. So especially when you're going around a cup and you're just then turning the paper as you're like slowly squeezing down on that. Where you go up to the top, turn, just rotate these around. That. Okay, scrap pile. And there you go. So cut out those individual blood cells, red blood cells, again, the antigens, the antibodies, and then we'll be good to go. All right, go Bears. Squad. Okay, so here's our scrap pile. We don't need that. Throw that away. We've got our antigens. We've got our antibodies. We've got our red blood cells. Then what next thing I want you to do is to take these that we separated and separate off the bottom here. Okay. And then go ahead and separate these just like this. Nice clean cut. You can also trim them up a little bit. Okay, so we've got that. These, what you can do is just leave them together for the moment. And so I'll go ahead and pause it, giving you time to do that. There's one other thing that I want you to do, and you're going to need your highlighters for this, and maybe some of your red colored pen. Um, and this is what we're going to do in anticipation for so this is what we're going to do in anticipation for the notes section. Okay, so we put that off there. I've got that here. I've got these over here. And taking your highlighters, what we're going to do is I want you guys to, there's 3 different colors or shades, I should say, of our antibodies. There's the white, there's the what looks like the gray, and then the black. So it can be a little confusing which one is white and which one is gray. So what I want you to do is you're going to just separate all the white ones out. Okay. Separate the black ones out, and then separate out the gray ones. So to make it clear, we're going to take the white ones and just color them the highlighter green. Okay. I don't know about you, but anything that will help me visualize and make very clear the learning that will be taking place is worth the extra time to do the setup. Another thing we're going to do is while we have our green in our hand, you're going to be separate out all of the B antigens. Mm-hmm. It's so fun imagining all of you guys do that at home when I go, mm-hmm. We really are a squad in class and out. And then with this one, just go ahead and color in the B with the green. Okay. Nice. Okay. So I've got my White one's colored green. I've got my B antigens colored green. Hopefully this is getting you a little curious about what are these antigens and these antibodies and what do they have to do with the blood as we're getting this set up. Okay, we're done now with the green, and with the blue, we're going to do the same thing, but this time with the gray antibodies. Blue. 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 And the blue, now, we're going to separate out the RH antigen. Antigens out. So I've got my little pile of A's. We'll just leave black. And now with these RH antigens, okay, we're going to color the RH in the circle blue. Now, the only other thing I want you to do is take your red blood cells. Okay. So with these, since they're red blood cells, you can take your just a red pen, and you don't need to color them totally, but just give them a little bit of love there with the color. Um, or you can even use the orange or red highlighter to just have a little fun, engage your artisticness, and get these all done. Highlighter is kind of like the shading portion, and then you can go back in. Hey, stop it. Hey, hey, you on your bed. And then you can stop it. Okay. Stay. All right. All right. So I now think that we're ready. We've organized all of our graphics into those piles. Now let's begin with our notes. Okay. What we see right here is blood after it's been centrifuged. So go ahead and write centrifuged. Magnify it for you. Centrifuged blood. Stay. Stay there. You stay. Right there. Okay. So I've got the centrifuged blood, and what happens is the centrifuged blood, um, blood is made up of what we're going to call cell components that are suspended in a serum or plasma. And what happens when you centrifuge, you spin it around at high speeds, is that it pulls all of the cellular components, because they are heavier, down to the bottom, and it separates the cell components from the plasma or the serum. And you wind up getting these three distinct layers. So the first layer right here is the serum or the plasma. I'm going to call it the plasma. Or serum. Okay. Then you've got the red color that we always associate with blood down at the bottom, and these are the red blood cells, also known as the RBCs. And then you have this distinct white little, it's very thin and narrow, this white layer between the plasma and the red blood cells, and that's the white blood cells. Also known as the WBC. Sounds like a TV company. Okay, white blood cells. And in any given sample of blood, you have approximately 55% of the sample will be that plasma, um, and then the rest of it will be the red blood cells, and the smallest amount will be the white blood cells. Okay, now what I want us to do is to take our highlighter, and with our highlighter, just to make it pop a little bit more, just go ahead and show that the serum is like this. Okay. So one of the things, Now, those cell components are floating around in that suspended in that plasma. And there are three different cell components that we're going to be looking at specifically. But since we colored our plasma in this highlight color, go ahead and just put this around to help us to understand that you've got those just floating around in that serum, suspended in that serum. So what gives the blood its red color really is the red blood cells, but because they're distributed throughout the plasma, the whole thing looks red until you centrifuge or you separate those out. Okay. Now, let's talk a moment about the plasma. Okay. Take your pen, and we're going to say now that the plasma something I want you guys to know about the plasma is that the plasma or the serum has two components to it. Okay. The main components I want you guys to understand is this is where we find the antibodies. And then just know that it's mostly water, but you have other proteins and glucose, sugar, also suspended in there. And our antibodies are represented by the kind of triangular shaped little graphic that we're going to be using. Got these parts right here now, when we're looking at over here, we're going to call these the cell components. Okay. And the cell component portion of our show, there are three basic things. Okay. You've got the white blood cells. So we'll put here, and I'm just going to, we know it's in the blood, so you just put white cells. Okay. You have these little guys here, which are your platelets. Bear, on your bed. On your bed. Plate. And we have our red cells. These are our cell components. Okay. Okay. And then All right. Here is our little something that I want to just give you some more specifics on the blood. Here we see the the centrifuged blood that we put in, the plasma 55%. This white composed mainly of the white blood cells, and then the 40. So this is a very, very small percentage of this, like 5%. And then the other 45% would be the red blood cells and the platelets. And then I like this graphic because it shows you that if I have whole blood, um, that 8% of the whole blood is really everything here, and then other fluids and tissues make up 92%. So of the whole blood, you've got that 55% plasma. This breaks it down more specifically. If you were taking an anatomy class, you would probably have to know all of this stuff. 7% of the blood plasma is proteins and albumin, globulins, fibrinogen, all others. 91.5% is all water. Helps explain why you keep hearing that, most of us is made out of water. Well, you'll find that in all of the body tissues and anatomy as you study. And then you have other solutes, which are the electrolytes, nutrients, gases. Electrolytes are what you're always hearing in those energy drinks or things like Gatorade. They're saying you're putting back into your body just certain chemicals that your body needs, regulatory substances. And then there are waste products that are going through there as well too, filtered out by the filtering systems of your body. So that's the plasma. Plasma now is when they say solutes. Solutes are things that are dissolved in the solvent. So when you take water and sugar and you put sugar in and you mix it up, the water dissolves, breaks apart the sugar into simpler components and dissolves it in the water. So the plasma, the water, portion of this is the solvent, and these things are dissolved in that plasma. Then you've got what are called the formed elements or cell components, we're going to call them. These components are formed in the red marrow of your bones. That's how they then get into your blood. And we're broken down into the platelets that we saw here. Uh, Man, who took the time to count how many platelets are in a sample of blood? White blood cells, and then your red blood cells. Of the white blood cells, there's specific ones, the neutrophils, lymphocytes, monocytes, eosinophils, basophils. As we're going to see, the white blood cells deal with your immune system, and you'd learn more about the specifics of what each of these white blood cell types do. So now looking more specifically, we've got the red blood cells. So here is a scanning electron microscope view of blood that has been magnified 3,000 times. You can see the red blood cells here. You can see the platelets, and then you can see the white blood cells here. So the red blood cells, the reason why they're red is because the red blood cells carry around oxygen to all the cells of the body, and then they transport the waste gas, the CO2, back to the lungs, where then they're outgassed when you breathe out. And you can see that they have a very distinct shape, look like a little inner tube that's not all the way through, but has this concave section in the middle and this chubby little section here. The reason why it's shaped like that is that there are certain vessels in your body that are so tiny, only one of these blood cells at a time, red blood cells, can go through. And the red blood cells, because they're shaped like this, can kind of almost fold up on itself to squeeze through the smallest little vessels that you have. What's interesting about these is that there's no nucleus. So kind of a trivial pursuit question, what is the one, what are what's a cell of the body that is non-nucleated? The red blood cells, they're so specialized to carry the oxygen that they don't have a nucleus. The only thing that they do is carry that oxygen and then the CO2 around in the system. And what enables them to do that is they have a hemoglobin molecule, and this is what it looks like. It's made up of 4 different proteins that are folded together. And there's an iron um, component to the hemoglobin molecule, and that acts like a little oxygen magnet. So when the red blood cells are going through the capillaries of your lungs and you breathe in, the oxygen that's in your lungs now sticks to that hemoglobin. And now in that little hemoglobin backpack, you can think of it, is then carried to the tissues of the body. Um, there are a lot of them in there. They do have a lifespan, and I believe it's the liver that filters out the red blood cells, but you might want to check me out on that. And so they're replenished every 120 days. That's why when you give blood, your body will rejuvenate that, but you can only give so much because you don't want to give so much that your body doesn't have enough of them to do what it needs to do. And I believe 120 days is the amount of time between when you can give blood. So that's the red blood cells. Then you've got the white blood cells. Um, these do have a nucleus. And so when you get DNA from blood, you're not getting it from the red blood cells, it's from the white blood cells that are present in the blood. Um, don't have any hemoglobin because they don't carry around oxygen. And there's 2 principal types. Again, when you study, we were to go back here, you see there are ones that are granulated, meaning if you look at them under the microscope, they are more smooth looking. And then there are ones that look like they have these little granules all in them. So they separate them into those 2 types, granular, agranular. This would be like one of the granular types. They live only hours to days because they're involved in the defense of the body, so they're working super hard. And so the lifespan is much shorter. Combat inflammation, also infection. And then we've got the platelets. These are like cell bits, and they're involved in stopping, kind of forming this fibrous barrier that when you bleed causes that blood to clot and keeps the blood inside your body. So you can almost think of it like almost a repair fibers that when you get a cut and you're holding a bandage or something over that cut, those platelets will accumulate around there, and the scab that you have when your body is healing is all the dead platelets and red blood cells and things that are now causing or forming that cap, that protective cap, while your body heals underneath that. Okay. Again, they're manufactured also in the bone marrow, have only a 10-day lifespan. Okay, so where does that leave us in our note situation? To really understand blood typing, which is what we're all about, that's just a brief background. You don't need to know any of the specifics about that. What I want you to know is that blood is made up of plasma, and then cell components or formed elements, they call them. And of those formed elements that float around in the plasma, you have the white cells, you have the platelets, and then you have the red blood cells. Inside of the serum, the plasma, you have those antibodies. That's going to be crucial in us understanding how blood is typed. And the red blood cells here, okay, there's something that you need to know about the red blood cells. The red blood cells, this right here, okay, as well as having hemoglobin on the surface, that binds with the oxygen, they also have proteins on the surface called antigens. Okay? And there are three types of proteins that might be embedded on the surface of those red blood cells. You've got the A antigen, you've got a B antigen, and then you've got a antigen that they call the Rh antigen. Okay, so those are the three antigens that are on the surface. Okay, and it's those antigens on the surface of the red blood cells that determine your blood type. Okay, so take your glue stick, put that on there. Get a little glue on there. So when you are asking yourself, these are blood types. What is it that makes somebody, what is it that makes the blood types different? Well, if somebody, it's all what blood type you have is determined by what kind of protein antigen you have on your red blood cells. Okay. So here are the, just do a little bit of graphic now of this. We're going to take one of each of our antibodies here. We're going to take one of our red blood cells, and we're going to take one of each of our antigens. Okay. Just like this. Okay. And what I want you to do is, because this is going to come in later when we're doing some of the activity here, you're going to take, and this is where, make sure you have something that you can paste your uh, make sure you have something that you can do your pasting on so you don't get the glue all over your book, right? So we're going to, like that, just put one of these here. Okay. Take another one. Put another one here. Oop. There. Then next to that, you to take your red blood cell. Or your 2 red blood cells. There. And then sometimes when it's something a little like this, I'll just put it on the top of this. So I've got the B. And RH proteins. So antigen is a fancy word. It's just a protein. Proteins are made up of amino acids. You'd learn all about that in biology if you've taken it. And then these, we're just going to go ahead and say that these are these are bodies. Where are they found? In the plasma or the serum. Antibodies. And this is going to be just abbreviate it, anti-B. This is anti-A. And anti-RH. Okay. And so here are our the different antigens. Okay. All right. Now what? So we're done with that page, and I want you to go on to the next page. And on this next page, we're just going to do some setup for work that we're going to do in class. Okay. And all right. And this is where your red blood cells are going to come in. Okay. First, we're going to set up some columns. And sorry, it bled through there. I shouldn't have used the Sharpie. Live and learn. So right here, we're going to put on this first column, go ahead and write RB C's. Okay. So that first column is going to be the red blood cells. Then we're going to write the plasma for the serum. Plasma. And then right here, we're going to put blood type. So the first thing we're going to do is we're going to identify each of the blood types. what does the blood look like? What kind of antibodies are in the serum? What kind of proteins are on the surface of the red blood cells? And so we'll look at that with these three columns. So let's just take one of the red blood cells again. And put it right here. Okay. Take another one. We're going to have 3 rows per page. Good. Yeah, that. Now, I don't think we need to make the headings again on the next page. You can just put the red blood cells so we know that to the far left here, this is going to be our red blood cell column. And here, just like this. Okay. And make a third one. Like this. Okay. 1, 2, 3, 4, 5, 6, 7, 8. Like that. And then on the next page, okay. Now, it looks like I didn't give you enough red blood cells if you're like me. So just take your red pen, pencil, and let's make let's just make some red blood cells there with the something like that. All right. So how this is going to work. We're back here. And with the background information that we have, we are going to go, and there's 1, 2, 3, 4, 5, 6, 7, 8 blood cell types. We're going to look at the anatomy of each of those blood cell types. Anatomy is what are the body parts versus physiology. How do those body parts work? We'll look more at how what's the working of the body when we talk about blood typing. But for right now, to just get us into the anatomy of this, let's start with, well, let's just go straight across. O positive, okay? So if we are an O positive, what that tells us is that we don't have B. We don't have A. Positive means the RH. We do have +4, the RH antibody. Okay. Um, Now to illustrate that, we've got a red blood cell. So you're just going to find your RH. You're going to take your glue stick. And we're just going to put it on there just like this. Now, obviously, there's not just one RH antigen on a blood cell. They're covered in them. Just to illustrate the fact that you've got that. So here, our blood type here, then there's no AB. So the blood type is O. And it has the RH factor, so it's O positive. Okay. Now, what I want you to do, let's just do one more. Make sure you understand this, okay? So that's the O positive. A positive. All right. So if you have A positive blood, what does that mean? That means, so we'll go here and we just go A positive. It's my blood type. That means now I've got the A antigen, A protein on my red blood cells. But it's positive for, meaning I also have the RH antigen on there. So I'll get an RH. And put it on there too. Okay. So your job, and then you're done. I was going to give you more, but we'll do it in class. So the next anatomy is going to be the anatomy of B plus. Somebody with B plus blood, what do their red blood cells look like? Then you're going to do AB positive blood. AB positive blood. Then you're going to do O negative blood. Then we're going to do A negative blood. Followed by B negative. And then the last one, AB negative. Okay. Now, leave the space still in the middle because that's where we're going to look is what's going on. If I have AB negative blood, if my red blood cells look like this, what do I have in my plasma? What antibodies do I have in my plasma? So get that all done. I'll expect that that'll all be done. We're not going to have time to do this. If you don't have time to do it, then come in at lunch on Wednesday. Then come in at lunch on Wednesday and get it done. But we're going to dive right into what's in the plasma, and then you're not going to be able to focus on it because you're scrambling trying to get caught up in the other stuff. Okay. So get that done. Any of the pieces that you still have remaining, be careful not to lose any, and put those back in your pocket in the back. and then we will be good to go from there. I'm not going to turn on the lab yet, um, so don't worry about that. Just do all of this work in your book for tomorrow. And stay warm and cozy out there. Get your homework done. I know there are projects coming up for your final big projects before the Christmas break. So break them into small chunks so you don't procrastinate it and it becomes a big problem later. And see you tomorrow. Go squad, go squad, go.